Welcome to our last Microbiology Journal Club Summer of Genomics book club. So this is our August book club. It's our third book club, and it'll be the final one for our Summer of Genomics book club that we've been doing. If you are catching up with this as our last one, this is the book we're talking about today. It is called The Society of Genes. It's one of our shorter books for this book club. I think it was a nice, friendly one to read in August as everybody is getting ready to head back into the semester in one way or the other. And so I'm looking forward to talking about this. Before we get started, I will do quick introductions. So my name is Laura Williams. I'm an assistant professor at Providence College in Providence, Rhode Island. And I will then turn it over to, I think it's actually Sangeeta who's next to me, if you would like to introduce yourself.
Hi everyone, I'm Sangeeta. I work at Imperial College in epidemiology, and my research was related to phylogenetics, so I'm very interested in all things related to gene. So that's why I'm here with the book club.
Excellent, and thank you very much for joining us. We really like your profile pic, so that's a good one to have up there. And next to Sangeeta, we've got Nicole. Nicole, would you like to introduce yourself?
Yep. Hey, everyone. I'm Nicole, and I'm a postdoctoral fellow at the University of Northern British Columbia in Prince George, British Columbia, Canada. And I've been working on microbiome methods to sort of look at forest soil profile disturbance regimes. And again, I'm really happy to be here to talk about another genomics book because I don't read enough about genomics, and 2017 has been a summer when I've done a lot of that. So it's been awesome.
And finally, we've got Joel. Would you like to introduce yourself?
Sure. I'm Joel Graff. I'm from the Montana Tech in Butte, America. And this is the first book club I've been able to join in on this summer. The first book, I read it and I was on vacation. Second book, I didn't like, to be honest. And then this book, I found it fascinating, but not quite finished, but close.
All right, and I like your fish back there in the background. That looks like a nice office companion to have.
Yeah, I have a new office, and the previous occupant said, "Hey, do you want my fish?" And I'm a pushover. Yeah,
never turn down an offer for free fish. All right, so thanks to everybody for being here. I'm really excited about this. So I thought what we would start is to talk about just general impressions of the book. So I'm going to give a really brief for people that haven't read it or who are maybe watching this because they're interested in reading it. I'll just give you a brief rundown of what the book is about. So when I wrote the description for the website, I hadn't actually started the book. So I just pulled something from the book jacket itself. And I think it kind of doesn't, the book jacket I don't actually think does great service to the book itself. It kind of talks about this idea of genes being a society and each of our genes in our human genome being a member of a society, and that's definitely the theme of the book. But this book touches on, each chapter touches on a different topic that to me is kind of like one of those things that if you are out in a group and somebody finds out you're a biologist, it's kind of like each chapter seems to be one of those things that somebody might be like, I heard this thing about CRISPR. What's CRISPR all about? And then there's basically a chapter in here that's like, well, let's just walk you briefly through CRISPR, you know? So under the umbrella theme of thinking about each of our human genes and our human genome as a member of a society, not only in our bodies, but also as all the genes that are in the human population. Under that umbrella, this book basically talks about a whole lot of interesting biology concepts. So we get, we cover immunology, both from the bacteria's point of view in CRISPR and from the human immune system's point of view. We talk about, what's the purpose of sex and recombination. We talk a little bit about human diversity and human evolution. So the idea that, we're only 0.1% different from each other. They kind of walk through what does that mean when we're looking at the human genome and thinking about similarities and differences amongst different people. And then they get into things like human evolution with chimpanzees and primates and leading to different lineages of hominins. And they start into all kinds of interesting stuff about gene duplications, gene families, freeloaders. So there's a lot packed into this book, but I felt like it was really dealt with in an accessible way. There were parts of it that I was familiar with, and I was kind of nodding along going, yep, that's familiar. And there were parts that I was less familiar with. So the last chapter, which talks about lines and signs in the human genome, was something that I'd heard of, but I was not very familiar with. And so I felt like I was learning a lot by reading that. I kind of wish we had a layperson, a person who's a non-scientist, who could read a book like this that's meant for the general public and kind of give us a perspective on what worked, what didn't work, because all of us have enough of a background that I think we're too far removed from that at this point to kind of know how it lands with the general public. But I'll turn this over to you guys now. What did you think about this book overall? And do you think they were successful in getting across all these kind of big concepts in biology? Go, Nicole.
So I have to admit that I think about a third of the way through the book, I wasn't really sure what to do with the extended metaphor of society in the book, because there was part of my brain that was just going, is this just a veiled sort of way to say that we are looking at processes involving genetic material and in some cases transcript material as context dependent, depending on endgames, which I mean, really, when you come back to it, societies we know are an assembly of people, but there are always intentional and purpose-driven processes that happen, and the mechanics of how they come together still have some recursiveness in terms of what humans do to cooperate and collaborate or to cheat. And I think in the end, I was convinced that based on how the authors also interwove chapters to bring back, you know, paradigms, or well, not necessarily paradigms, but sort of mental models that they presented in earlier chapters to say this new biological snippet of something happening is either a variation on this or is taking these concepts. So there was a lot of intentional design in the textbook that I think by the time I got to the end, I realized why this actually was an excellent piece of science education writing, because it's a lot of work to be that intentional, and that intentional when it's a book driven by very intricate conceptual models and qualitative descriptions of processes, and trying to find, you know, I mean, these are metaphors that take up almost half a chapter or an entire chapter. And some of them are quite unconventional, like the kibbutz versus the Moshev comparison for diversification of function. But I mean, it's a really interesting way to talk about stuff that from a protein biochemical perspective, you know, we never talk about it in terms of strategizing how we diversify function on an evolutionary landscape. It's always very looking at minute structural parts. And I think that was the other interesting thing for me as well, is that because we are all not lay people, we were also used to the paradigm of a lot of strategies of gene expression and gene diversification always being accompanied by the visual and the technical talk about the biochemical elements happening. And that was also stripped away, in addition to focusing heavily on history of genetics, which is something that happened in Siddhartha Mukherjee's book, The Gene and Its History. So I'll just leave it at that for now, because I'm saying too much, I think.
Well, I'll chime in. Sorry, Sangeeta. I was just going to say that I felt like this book, it started talking science and it never stopped. And so like in the Siddhartha Mukherjee's book, he would be talking science, and then he'd have kind of an interlude where he talked about his family or whatever. Because I wanted to just be about the science, I kind of didn't like the stepping back and looking at big picture of family in the Mukherjee book. And so I appreciate that it was just science, science, science. Now they used a lot of metaphors and analogies to get the science by, and so then I think that's how it kind of ties into working as a popular science book. But I worry that maybe if you're not a super nerdy person like me, that the consistent science and never giving you a breath might turn some people off. And then I teach evolution, and when you're talking evolution, a lot of times people talk about evolution on the population level, or they talk about it at the genetic level, and they're usually talking one gene at a time. You get a mutation that causes one change or whatever. And the fact that multiple genes work together is something that that maybe I haven't ever read. I shouldn't say this, but I haven't ever read The Selfish Gene by Richard Dawkins. But there it was talking about how genes, it's at a gene level that you should think about evolution, is my understanding of that book. But this talks about the interaction between genes and gene networks, and I thought that that's important, because when you hear about Gregor Mendel's research, for example, he followed these traits that were all controlled by a single gene. Whereas now, and there are some genetic diseases that are caused by a single gene, but most of them turn out to be far more complicated and involve far more genes at a time. One of my favorite analogies that they used in the book was the lock and key analogy, where if you have a key that fits a lock and you change the key a little bit, it'll still probably work, but it might not work as well. However, if the lock causes there's a change in the lock that then matches up with the new version of the key, you're back to working great again. And that's just a two-gene interaction sort of an idea, but, well, I guess unless you think of each sector of the key is. But anyway, I thought that was maybe, I got to about 55% through the book. I thought that was maybe their strongest analogy or metaphor based on the title of the book. But anyway, like Nicole, I'll stop talking now. I'll let Sangeeta jump in.
Okay, so in contrast to everyone here, I guess I did not like the book. I'm about halfway through the book now, and I did not like the book because I think there was a lot of condescending tone. I thought the language was a bit condescending, in the sense that, for example, if you look at the pictures that they have made in the book, they have like a smiley for a virus or something, which is, I think, very juvenile, and someone who would pick a book like this to read. I go back to the days before I did science. I would have picked up a book like this, and this would have definitely annoyed me, and it annoyed me today, even when I know those concepts. So I think Laura, you were talking about using this as a kind of a textbook or sharing it with your students, but I think your intelligent students might resent the tone of the book. So that's why I was not able to finish the book, because I was not enjoying it. But yeah, the idea of the interaction of genes, and I think, and I also like the lock and the key metaphor very much, so that made it very clear. But other than that, I'm kind of not very enthusiastic about this book.
Yeah, I think that's an interesting perspective. So that's why I'm always glad when we have multiple people who come to the discussions, so that we can get a range of perspectives on this. I wonder, I feel like they were kind of trying to walk a line between writing something synthetic that their colleagues would want to read and go, oh yeah, that's really interesting that they're kind of pulling all these ideas together under this larger thesis of the Society of Genes. But I think they were also maybe trying to write something that they felt was accessible to the general public. Sangeeta, do you think that it would have made sense for them to just kind of lean harder into one of those ways, or do you think that do you think they were oversimplifying things that they maybe didn't need to?
Yeah, that's what I think. I think they have underestimated the intelligence of the general public. And if you look at other popular science books, they deal with, for example, Siddharth Mukherjee's book was not specifically aimed at a scientist. It was aimed at general public. And a lot of people have read it and they have enjoyed it. So I think all the metaphors, etc., are fine, and I don't really think that. Yeah, well, that was also an issue. I think they were trying to oversimplify it and explain it in a way that you would explain it to a kid, but a kid wouldn't pick this book up, and the person who's picking up this book up is not a kid. So there's that thing. So I think the language could have been more taught, and they could have introduced more terms, and people would have been okay with that, and less fancy pictures. I reckon that was one really annoying thing for me. So yeah, the focus of the book could have been clearer, and while catering to a scientific audience, scientifically literate audience, not specifically scientists, and introducing this idea of the Society of Genes, I think it could have been a much better book.
To go along with what Sangeeta is mentioning, I felt like after the first 3 chapters, I was having that same feeling. I thought that it kind of started off a little bit gimmicky feeling. However, because I suggested this book for this book club, I stuck with it and tried to get as far as I could, and I felt like the tone got better over time. So yeah, I think it started off pretty basic, and then by the time you're to the 5th or 6th chapter, it's getting a little bit more complex, and I'm not, if someone can handle the 5th and 6th chapters, they're going to have trouble getting through the first couple. If someone finds those first couple chapters just the right tone, they might be in over their heads by the time they got in a little later in the book. So it was a little uneven.
Yeah, I think, Joel, I think that's I hadn't really thought about it in those terms. I was kind of taking each chapter and thinking about each chapter individually, but I think that's a good point. I do think that, so one of the things that I am planning to do with this book is so I'm teaching genomics in the fall, which is part of the reason why I chose the theme for this summer's book club. And so what I'm going to do with the first chapter, because the first chapter basically uses cancer as a framework for introducing some topics, some basic topics and concepts in biology, cell biology, and genetics. So my guess is they thought, okay, since we're trying to aim this at as wide an audience as possible, we want to bring everybody up to speed in case they're sitting down with this book and going, you just said chromosome, and I vaguely remember what that means, but I don't exactly remember what that means. I think that's what chapter 1 was about. And so what I'm planning to do for my students when I teach genomics is we're basically going to have one class session very early on where they're going to read the first chapter, and then we're going to walk through it in class, and I'm going to ask them to overlay what they've learned from Gen Bio, and if they've taken genetics, which many of them haven't, but they've taken a course called Cellular Molecular Biology. I'm going to ask them to basically go, as a class, we're going to go through the first chapter, and they're essentially going to annotate it with the more advanced language and topics that they should be able to pull from what they've learned so far. Because they don't, as both Sangeeta and Joel have mentioned, they kind of dispense with the official language of these. Like, at no point do I think they say transcription. They're very they're trying to kind of, which I respect. I mean, I think they made a choice to say, we're going to try to introduce minimal jargon. So we're going to talk about these processes, but we're not going to introduce all the terms that we're used to using. We're not going to, in some, they talk about non-synonymous and synonymous mutations, but they don't call them that. One thing I thought was interesting that I think they left out, I don't think they ever talk about introns and exons and splicing, unless I missed it, which I kind of got to the end of the book and I thought, oh, wait, because I work with bacteria, so we don't have to really think about that. But I was like, oh, wait a minute. They didn't talk anything about the fact that there's splicing that goes on, the fact that a gene itself in a human genome is actually made up of these very distantly spaced parts that actually code for something that has to get processed. I don't think they ever discussed that. So there's some things they chose to skip, which, to Sangeeta's point, maybe they should have discussed. But that's what I'm planning to do with chapter 1, because I do think it's an intro that, as you guys have mentioned, if you've got some background in biology, you should be able to just sail through that and go, yep, that. But I'm hoping that this is a good refresher for some of my students if, you know, their Gen Bio knowledge is stacked behind all their Game of Thrones trivia knowledge somewhere. Yeah, those are interesting perspectives. I had a couple other things, but I didn't want to move on unless somebody, in case somebody had something else they wanted to add.
As long as we're talking about chapter 1, I was just going to say that I was happy that they decided to talk about the hallmarks of cancer, that Weinberg and Douglas Hannum, maybe, they made a pretty famous Cell paper about 2001, 2002, and then revisited that about 10 years later. And I always thought that if I teach a cancer biology class, I would structure my class based on those hallmarks. So that's it.
Yeah, I liked that too. I actually, to be honest, I didn't know about that. I hadn't read that paper, and this is the first I had ever seen, because again, bacteriologist here, bacteria generally don't get cancer. That's the first I'd seen of like, here are the steps that need to happen in order for cancer to emerge. I hadn't actually read that much about cancer biology to know that topic. So I think they chose the right framework, because this is something that a lot of people are going to have some type of personal relationship to, either themselves or someone they know. And so that in a way you're kind of learning about what's going on in your cells by virtue of when it goes wrong. Yeah, that was interesting. I hadn't heard that before. Other thoughts about chapter 1? Okay. All right, so I have, one of the things I wanted to ask for folks is throughout the book, they actually do, they pay a lot of attention to the idea of natural selection. And I thought this was an interesting feature of the book, where they are trying to explain natural selection and evolution, but they do it on multiple scales. So from chapter 1, when they're introducing cancer and they're talking about mutated cells. So cancer is a mutated cell in your body that's going to be dividing more rapidly and not undergoing cell death. And so they talk about it in the context of, well, this is natural selection at work in your body, because your cancer cells are going to be dividing more quickly than your non-cancer cells. And so they have higher fitness. Their mutations are heritable because each daughter cell gets the mutation, and there's basically, they're ticking off the boxes for having natural selection. And then they take this all the way to the scale of looking at the way we're familiar with it on the scale of populations. So selection on organisms' fitness. So not the cells in your body, but you as a whole assemblage of cells and your reproductive fitness. I just wanted to ask how you all thought either the small scale stuff, or, you know, if you got through all of this in the book, just how they treated it broadly. Did you like the way they treated that? Do you think that they did a good job getting across the concept of natural selection, or was there something from that you might have changed to try to address that topic more thoroughly?
I think I'll start, because based on my training, I have not taken courses that were very natural selection centric. And I actually think I appreciate what the authors did with the book, because I mean, they introduce those 3 prerequisites: the variability, the heritability, and the filter, the ever so contentious filter. And I think the cool thing about doing it on different scales is, for a book of more general readership, or whoever we assume is, you know, not coming to this book with the same knowledge bases that we have, not necessarily less advanced, is that, and showing this plurality about where selection comes and how it plays out depending on the biological context and motivation. Scale is a big part of that biological context. So the fact that they were able to be recursive and say, look, even though bacteria aren't doing you know, the paradigm of sexual reproduction for partitioning out their genetic material, you still have filters when you've got horizontal gene transfer, which of course, I mean, we know with respect to resistance genes. So showing that different configurations of genomes are still subject to undergoing the rules of natural selection, which is the easy one because things are falling out of my brain and I can't think about other things. But even in terms of testing the VDJ combinatorics for the immune system, so making sure that they're not attaching to your own cells and triggering autoimmune responses. So these editing functions, so who gets to pass? Who gets to go on? Like in this test of fitness, whatever this test of fitness is, I think they actually brought the best of both worlds in terms of giving people, you know, the bulleted list of the tenets that make this selection what it is, and also showing that it doesn't look the same depending on what frame of organismal or cellular or genetic material interaction that you choose to look at in each chapter.
One thing I thought was interesting is that since they started with cancer and they talked about how that's kind of analogous to natural selection and you know, the cells that grow best and increasing their chances of collecting more mutations and that sort of thing, then they kind of pull the rug out of that and say, but cancers don't go from generation to generation. So I just told you all about this, and then that's not evolution. And then they get to the next chapter and they're talking about B cells and how the B cells can, you know, they have maybe a weak interaction with their target, and then they go through a process of becoming better and better at interacting. And then they go, but that's not really evolution because those don't pass on through the germline. I thought it was kind of funny, but I also wondered if someone that was reading this is like, when are you going to get to the Society of Genes and evolution? They gave you kind of two really interesting things to look at, and then they pulled it away saying, no, that's not long-term evolution, as we're going to eventually talk about.
Yeah, that's true. They did. I like how they tried to fit that into the idea of just what are we talking about with natural selection, but then they are, you're right, it is the pulling the rug out from under you to basically be like, but that's not probably what you're thinking about when you think about evolution. Sangeeta, did you have a thought on that?
Yeah, I also like very much the idea of eliminating the idea of natural selection via cancer cells. I think it's a very good, and as you mentioned it, you can see it going on two scales within your body and then with the Society of Genes. I think that's a very good way of eliminating those concepts. I liked it.
Yeah, I think that's a good, because I think that's a complicated concept to teach. And I think, you know, as an educator, I think that kind of it's such an important topic that I think students hear it and then they're like, yeah, that natural selection thing, I know, Darwin, whatever. Some Wallace guy too, probably, I don't know, you know. And so, but I think to actually realize the power of this idea and how it impacts cells, organisms, populations, species, life, I think getting it repeated in all these different contexts is a good way to help students kind of go, okay, now wait a minute, this is actually, it's not just an idea that's kind of floating over here in isolation, it's actually a through line for all of biology. And I think that's good. I think that's a good way to get students thinking about this concept, which is so, you know, essential to understanding a lot of different things about biology. Okay, any other thoughts about natural selection, how they treated natural selection in the book? Okay, all right. One of the other things I wanted to, and I think it was Nicole who already brought it up, is the idea of all these different analogies. And let's see, I think that was really an interesting part of how they were trying to explain a lot of different concepts. They really went for a lot of different ideas. I mean, you get 6 degrees of Kevin Bacon showing up in the book, so they're really going all over with these. I'm gonna skip a little bit over to the chapter that Nicole talked about, where she mentioned that they made a comparison to a kibbutz and a moshav, and I'm maybe very much mispronouncing those, so I apologize if I am. And they did that in a way of trying to talk about, it's a chapter that discusses how do you get novelty? So how do you end up with new functions encoded in your genome? And I actually think, and this is chapter 8. So it's called Theft, Imitation, and the Roots of Innovation. I actually felt like this chapter did a pretty good job talking about some very interesting ideas about where do you get genetic novelty, where do you get functional novelty in a genome. So they have another chapter where they talk about how you manage existing genes is really important in terms of tuning gene expression and when genes are turned on and when genes are turned off. But this chapter specifically is discussing the role of gene duplication and then the role of gene transfer. And so they've got this great analogy where they talk about, okay, well, if you're in a kibbutz, you have a system of collective living. And so every individual stays within that community. And if you have a mom who has 2 daughters, probably what's going to happen is the 2 daughters are going to kind of take jobs that their mom had done, and they're going to start to specialize. And so they use that as an analogy for explaining when you get a gene duplication, then what you might have is those 2 genes end up encoding proteins that start to kind of do slightly different things. And so then you get 2 proteins that had the same origin, but they've kind of changed and mutated, and now they play slightly different roles. Depending on if you've given a lot of time, they might play very different roles. But then they also talk about the moshav, which is a community, but it's a fixed size. So if you had 3 kids, only one of the kids could inherit the land, and so 2 kids are going to have to go off and do something else. And so there's basically a transfer of function to a new society. And then they kind of walk through, after they introduce that analogy, then they kind of walk through, they talk about hemoglobin, they talk about smell receptors, and they talk about opsins and Hox genes. And I thought this was great. I thought this was a really neat way to get across to people, and it's a short chapter, but to get across to people the idea of gene duplication and gene transfer as being ways that you actually introduce true novelty into the genome. I know a couple people probably didn't make it this far, but if you did, and I think Nicole did you have thoughts on this chapter and how they did that?
Yeah, I'm just kind of navigating back to that chapter now, and I think.
Yeah, sorry, I sprung that on you.
Yeah, no. The ideas of actually introducing duplication as one of the conceptual strategies I thought was really, really helpful, actually, because again, thinking about this from the educator perspective, physically, you can describe that in terms of what's happening in a genome. And translationally, you can describe that in terms of, isoform specific expression of protein. So there's all these nice things like you suggested doing with your class with chapter 1. Can you superimpose sort of the more biochemical or molecular biological things onto that? So they've done a really good job of like breaking this up, and then also talking about, the gradual trajectory of mutation within a gene that can either give you diversification if you've duplicated or just allow a new function to arise. And I thought, it's a small piece, but the thing that I really kind of appreciated was that they do address things like functional and catalytic promiscuity of gene products. So that's pretty elegant, because now we're getting into things that do get addressed usually at the biochemical level by looking at function. But also, I think it's important because the whole idea of using the Society of Genes analogy is about motivation and function. And as we see once we get to the last chapter about lines and signs, sometimes the only function is we're going to just efficiently make copies of things. But the idea of singularity of function is something that even as scientists, we want to try and reduce things. And like, it's the simplest model of correlation is we have this configuration. Does it have a clearly defined role somewhere? And I mean, crystallins are structural, but they're also doing lactic acid transformations. A lot of proteins have active sites that allow you to do 1 or 2 things depending on what's in the area, or depending on whether they're allosterically modulated. So it's actually a really elegant chapter for starting to think about the Society of Genes and then sort of the legacy of the Society of Genes, which is the products that are transcribed and translated, and how the Society of Genes, you know, set the groundwork for that to actually play out in a physiological context.
Yeah, and I think that's there were times that I was reading the book where I was kind of felt like I was working to try to figure out where the Society of Genes, where their theme was fitting into what they were talking about. But this chapter, as Nicole, as you just discussed, I think this chapter is one of the chapters in which they're making a real argument for expanding beyond the selfish gene. Which, Joel, I have read The Selfish Gene, but it was so long ago that I don't really remember it. I was reading this book and I was like, dang it, I wish I had read that more recently or had time to read it now. But yeah, but this chapter where you, and the ideas that you're talking about, Nicole, about promiscuity of the gene functions and the idea that they're the crystalline example. So you've got these genes that are encoding products that do two functions that you would not think are related to each other at all. But all of this is interactions. And so these couple of chapters towards the end, like I think it's also 7, yeah. So chapter 7, where they're talking about how managers, transcription factors, gene regulation, how genes are turned on and off, levels at which they're expressed can affect, you know, have huge differences. So this is the latter part of the book is when they really start to go hard into the idea that interactions amongst genes are incredibly important. And that's how you get the emergence of the organism. That's how you get the emergence of, you have a mouse or a human, you know, then why do you have these differences? And why does a fruit fly sprout wings and then you can get it to have antenna or legs coming out of its head? You know, why does all of this happen? It's because of all these interactions amongst genes. And I thought they did, and I thought that was well handled. They've got a nice, I'll just hold it up. They've got this nice bit where they, if you're into circuitry and electronics, they're like, oh, see, you got these circuits and they might be a fire alarm or they might be an AM radio. And I was like, gosh, I wish I knew electronics because I don't know what any of that means, but I see the point they're trying to make. But I think that was a good way of handling that without trying to delve too much into all of the very particular details of each one of the examples that they chose. I think they got just enough in depth into the examples to be able to have it feed the thesis, and then they moved on, which I think was an okay way to do it.
I just had a thought while you guys were talking about the gene duplication, and sometimes when I'm studying for a class, I often think, well, how would I teach it? And sometimes when I'm reading a book, I think, well, how would I write this? So while you guys were kind of explaining that chapter, one thing about gene duplication that I think is interesting is that in my research, we study a family of proteins that is quickly expanding. Certain families of proteins expand faster than others, and this family of proteins that I study, they're called TRIM proteins, but they're involved in the immune response. And so there's a pretty strong selective pressure on genes associated with immunity to expand quickly compared to other types of gene families. So, you know, I guess I would bring, if I'm talking about the Society of Genes, I might talk about families within the society and that sort of thing. But I guess they kind of got to that where they had the daughter with the 2 daughters that each specialized. But anyway, that's what came to my mind. But just the fact that you might have some gene families want to expand faster than others would be something interesting to add to a book like this.
Yeah, I agree. And I think they got a little bit into gene families, but I don't, and Nicole, you can correct me if I'm wrong, but I don't think I think that is part that they could have expanded if they'd wanted to, talking about gene families. Does that sound right to you, Nicole? I think they touched on it a little bit, but maybe not heavily.
Yeah, I mean, they got as far as saying that if you've got diversification of the gene, and there's some sort of traceable similarity, then yes, you're talking about a family. And I actually think that that's, from the educator perspective, that's actually a really nice level to leave it at, because I mean, there's different levels you can choose to teach that at, as whether these are gene families in terms of genetics structure, or the manifestation in terms of talking about it at protein superfamily level, where you've actually maybe got a topology that has existed, you know, in a genomic landscape for a long enough time that things fold up the same way, but they no longer really have high degrees of identity. So I mean, there's, again, these legacies of family-based trajectories of gene change that can put that family level at two levels, either at the gene level or at the protein level. And even then, once you get to the protein level, are you defining the functional family by symmetry of topology, symmetry of reaction? How strong is that? So I think they put it somewhere that left it open for people to understand that relatedness is an organizing principle. And I think that's also, again, an overarching theme with the book is there's these coarser levels of organizing principles for genes and their interactions, which is effective.
Okay. I'm going to skip now to the back, kind of sort of to the beginning of the book, which I think, chapter 4, which I think, based on what people have said they got through, I think everybody got to chapter 4. So I think that's like a third of the way through the book. So I wanted to touch on this chapter because this is just, I thought it had an interesting title. So they called it the Clinton paradox. I don't know if this is a term that they've invented. I had not heard that one before, but it basically has to do with the fact that Bill Clinton was in office when the Human Genome Project was wrapping up. And this is where you get the introduction of the infamous or famous 99.9% similar number. So this idea that people have probably heard of, that our students have probably heard of too, which is if you take any two people, they're going to be 99.9% similar if you looked at their genomes next to each other. And then they kind of walk through this idea in a way of looking at how genome variation in humans, basically. And one of the things they do at the very beginning is, which I thought was interesting, is they elaborate on that by saying, okay, yes, that's true. There is 0.1% difference, but that's basically if you're looking at SNPs. That's basically if you're looking at single positions that have mutated. But if you wrap in indels, that adds a little bit of difference. So really, if you look at the range of mutations, if you're thinking about SNPs plus insertions and deletions, what we're talking about is like 99.5% similarity between people. And then they have a bit in the next page where they kind of, I think it's a little longer than that, they kind of walk through how that number shifts depending on how closely related you are to somebody else, which I'd actually never read a breakdown of that before. So it's on page 102, and on page 102 and 103, they've got a little figure where they kind of draw. I was trying to figure out who all these people were because I figured they must be famous people. I was like, who are these people? There's like, you've got your really great like 70s hair guy there. But what they're doing there is they kind of say, all right, so if you pick any random stranger off the street and you're not related to them, you're probably 99.5% similar in terms of your genome. But then if you walk through your family tree, that number is going to change based on whether it's a sibling or a cousin or a grandparent or so forth. I'd not seen this broken down like this before. Had any of you seen it kind of reported in this way? Or how do you think that did you like this part? Do you have concerns about this part? Do you think it's accurate?
I hadn't seen it described like this, but this is kind of the turning point where I felt like they got a little bit more serious, and maybe it's just because they were getting to stuff I didn't know about, and so then it was a little bit more interesting to me. But you know, I teach meiosis. I think the previous chapter was on why you have sex, but so I've taught meiosis and things like that in evolution. But then they start getting to this comparative genome stuff, and that's a little bit out of my league, and so it was very interesting. And the other thing that they mentioned in that chapter that I thought was pretty good was that they, you know, if you think about the human genome's 3 billion bases long, and it's got a second strand, so now you're up to 6 billion, and then you say, well, 0.00 or 0.1% sounds small, but then if you think about it, it's 6 million, and now is 6 million enough to worry about? And then they go through the ideas that, you know, not all changes even mean anything. In fact, most changes don't really mean anything. Anyway, I thought that chapter was nicely done.
Yeah, I agree with you, because I had also not read this treatment given to this topic. So the different levels of differences, which was, I think, good. And this 99.9% thing is one of those headlines that, you know, that really annoys scientists, because it doesn't really mean anything. And it was good that they kind of explained why it doesn't mean a whole lot. And again, 6 million base pairs difference. And in addition to that, you have other differences. So to say that we are pretty much the same and then a little bit different, it doesn't have a lot of meaning. So I really like their treatment of this topic in this chapter. It was illuminating. I learned a lot of stuff from this chapter.
Nicole, did you have thoughts on that?
Yeah, I think I sped read this chapter out of frustration at 2 in the morning, and I have a little bit of an arithmetic phobia. So by the time they got to the gene accounting, I'm just like, this is not going to go so well. Because I mean, the last time I really had to think about something that looked like that was maybe acknowledging the one section in my high school biology textbook on coefficients of inbreeding sometime in 1994 or 1995. So it was kind of like this weird like reminder of how I didn't understand it back then, probably because I wasn't patient, probably because I studied at the last minute for everything. And strangely, I don't even remember seeing this in my 400 level human molecular genetics class, which I do recognize was a lot more focused on disease context for genes. So in that case, I mean, what you're talking about are, you know, differences in genomes that are functionally translating, or are differences that are linked to things that are functionally translating into differences. So I mean, the concept of difference and the effect size of those differences are quite distinct from what was trying to be advanced in the Clinton paradox chapter. So it actually, at some point, when it makes me less anxious, I'd actually like to revisit this chapter and see if I follow the logic a bit more. But it's cool. It's actually really cool to revisit relatedness about this and think about what does it really mean, as Sangeeta mentioned. But I also really like that, you know, based on some of the stuff that was mentioned in Fatal Invention, that they make, you know, appropriate weighted comments about how important isolation of populations are for creating human difference. And also a recurring theme in a bunch of chapters other than chapter 4 about single mutations that generally have no effect really do get swept along with the temporal trajectory of genomic change from generation to generation. So I mean, there are a lot of things that can just flip back and forth with no consequence, which I think does link to this in terms of impact of similarities and dissimilarities.
Sticking with this chapter, later on in the chapter, they talk a little bit about the amount of genetic diversity you have from the people that left Africa, a small group that then you get a fair amount of inbreeding going on, that left Africa, say 100,000 years ago. Meanwhile, the genetic diversity within Africa is huge. I have 2 biological children, and my other 2 kids are adopted from Ethiopia. And you know, being stupid Americans, we usually think, oh, Ethiopia, the only thing I can think about Ethiopia is that they are marathon runners. And so then people are always like wondering if my kids are going to be good runners. And, you know, I don't think they're trying to be prejudiced or whatever, but I just think they don't understand how much diversity is in Africa. I mean, in the country of Ethiopia, there's 80 different languages that are spoken, and I never left the capital of Addis Ababa, but the people that I was talking with that were from Ethiopia, they would be able to look at people and suggest what part of the country that those people came from. You know, the marathon runners being the tall, super skinny legs sort of people, that was kind of in the southern end of the country. And then in the northern end, you had kind of short, squatty people. And anyway, my two children come from roughly the same area within Ethiopia, and they look very different. They're not biologically siblings. But like, for instance, my son has super almond shaped eyes that are just amazing. And then my daughter, there's not a hint of that. But anyway, the idea, the thing they said that I thought was really kind of interesting later on was that, you know, perhaps the best of the best people at different specific activities are going to come from Africa once the playing field is leveled out. So, you know, maybe chess champions will be coming from Africa because they have so much diversity, but also some of the worst people at playing chess might also come from Africa. And I liked that they hit, you know, there might be some both extremes going on. And anyway, I just really appreciate when people try to get the word out or educate people about how much diversity is in Africa versus outside of Africa. And I'll stop there.
Yeah, I even, so this whole point about diversity within Africa, my reading of this chapter again tied into my reading of Fatal Invention, and I really liked the connections, and I understood more clearly what Dorothy Roberts was talking about. But there was one thing that struck me was where they mentioned malaria. And I vaguely remember that this idea that the sickle cell thing is commonly found in malaria endemic regions, that was probably challenged in that other book, Fatal Invention. So I somehow remember that, but it's such a common story that was the only time when I read Fatal Invention, that was the only time I had heard that story challenged. So I was not sure whether I remembered wrong or whether this book has somehow repeated that, you know, that wrong idea.
Yeah, I noticed that too. I was trying to find, as you were mentioning that, I was trying to find the part in the text where they talk about sickle cell and malaria. I couldn't find it though. I actually want to, similar to you, Sangeeta, I'd like to look into that a little bit more to kind of try to clarify the points that were raised in Dorothy Roberts' book with the example as it's typically given. Because if it's not an accurate example, I want to use it. Or if it's an accurate example in the right context, I want to make sure I'm presenting it in the right context. So I agree with you that that's a point. I made a note of that when I read that part too, and then I didn't have a chance to actually follow up on it, but that's something that I'm hoping to do before I, you know, teach something like that.
So one of the reasons I let off saying I didn't like the book from last month, and perhaps I just didn't give it enough of a chance, but I felt like the idea of doing anything medicine based on race or whatever is just completely invalid, was kind of the feeling I got from that book. And then I just, you know, mentioned about how much diversity there is in Africa. If anything, we need to do more understanding of how drugs and therapies work with our genomes and personalized medicine. I'm kind of a big fan of that idea. I understand you could hit some landmines maybe of people taking out of context and thinking there's superiority over one versus another set of genotypes that are out there. But for example, my kids coming from Ethiopia, something that happens in Ethiopia is there's a high percentage of people that have a different variation of a cytochrome 450 protein, and it makes them metabolize codeine very quickly. And so if my kids were ever to get codeine, there's a higher than typical chance that they would metabolize it too quickly and might overdose on morphine. So to me, that's good to know. And I just kind of felt like from the, as much as I read out of the Dorothy Roberts book, I felt like she was fighting against that. And I guess, you know, that's why I stopped. But since you guys read the rest of the Dorothy Roberts book, maybe you can correct me if I got that wrong, just based on the beginning chapters.
I think that what she argued was actually that I don't think she's against the idea of looking at genomics in terms of its intersection with medicine. I think what she was arguing is that the way that we're currently using race to try to, as a proxy for trying to understand what people's genomes probably contain, is just incredibly poorly defined and kind of useless. So to use your example, when your two kids get older, so you have 4 kids, 2 of them are your biological kids, 2 of them are adopted. As your kids get older and then they get married and then they have children. So when your children from Ethiopia get married, and perhaps they'll marry a white person, and then their kids have to make a decision about how they're going to consider themselves. So it's possible that they might classify themselves as white. So if they went into the doctor and they said, oh, you look white to me because you're very light-skinned and you must be white, so I'm not going to test you for all these things because you're probably white. But they might have inherited the allele that you're talking about that they then wouldn't get tested for because they didn't look not white, and maybe they decided not to report themselves as having Ethiopian ancestry. So her argument was basically to say, there might be some connections between people's history and their likelihood of having a particular gene, but the way we classify people, the proxies that we use, the way we group races together is so crude that it's not worth trying to use these categories as a real way of doing medicine. I think that's what, I mean, Nicole, you read the book as well, and Sangeeta, it sounds like you did too. So you can chime in if you think I've misrepresented it.
Yeah, I think you're exactly right. And I think, Joel, probably if you gave the book another chance, you would probably like it, because I think the author's point was exactly what you're saying. So what my understanding of the book was that she was saying that the genetic clusters do not overlap with our understanding of race, the way we use the word race. And that's precisely the problem. So while there might be a particular allele found in a genetic cluster, a lot of, for example, as you mentioned, a lot of Americans just think that whole of Africa is just one place, and they do not appreciate the genetic diversity there is in. So there might be an allele that is probably not present in people from Ethiopia, but they would still be lumped together in that category of American African and be treated accordingly. So because those two categories do not overlap, and we use the social construct of race instead of using the genetic clustering, which is a much precise tool, and the social construct is a very blunt tool which is being used, and it's causing more harm than benefit. I think that was her point. So if you read the book, I think towards the end I really liked the book, and it was quite thought-provoking. I realize it's discussion is digressing into the discussion of the previous book, but it was a very good book.
Yeah, I don't mind at all that I think we're and as we mentioned, we're in a chapter that the book that we read previously and the chapter 4 and a couple other places in this book, I felt like they aligned really well. And I actually appreciated seeing these authors. It's clear that these authors have knowledge of these ideas. It's clear that these authors are working to present these ideas as well. And it made me glad to see that. Nicole, I wanted to give you a chance if you wanted to say anything, and then I wanted to see if Joel had any follow-up to what we just said.
Yeah, I'm going to try to make this brief, which always seems to backfire, but I think I agree with both what you said, Laura, and also what Sangeeta said about Dorothy Roberts' criticism of how the categories created by the social construct definition of race introduced at the beginning of Fatal Invention are being superimposed as sort of the covariates when we look at genome-wide association studies or genetic, genetic data. So I mean, there is that exploration of the idea of thinking of ancestry, which is a bit more in line with what we are trying to sort of deal with when we're using genetic data in a diagnostic context, which is that we know that genes are heritable. We know that things like these P450 alleles are traceable based on relatedness to other people, but we need to call the stratum something different other than race, even though, there's still the chance that there's some very loose or crude overlap on those categories in terms of how alleles are distributed or your chances of inheriting them. So it's interesting that you bring up the P450, because I think 2 or 3 weeks ago, I actually saw a thread on Twitter by a Black woman who had probably the same allele, and ended up in an emergency room where they weren't responding to painkillers at all because of this metabolic profile. And it was an opportunity for this person to talk about how race and ER admissions, that this is one instance where that misconception, the racist misconception that Dorothy Roberts also brought up in the book, that Black people are somehow more impervious to pain, has translated into assumptions made at ER admissions to not give Black patients certain types of analgesia. So it's definitely an interesting complication here, because we've used this bad stratum of classifying people by race that is now making interpretive decisions that are ignoring actually the strength of genetic data, which is that you do want to consider something, a heritable metabolic quirk of your liver enzyme profile. That is something we should be screening for in personalized medicine or assessing personal risk. But the categories that we've decided to use to put a frame around the data, I think, is what Dr. Roberts is saying is wrong. And it's still complicated to figure out what we're going to do with ancestry or categorization, because people do move around and the interpretive framework for that are different, but it doesn't change the fact that such strong functional correlates to a genome are things we should be screening for. We just have to find a better way of discussing it in terms of a way that's inclusive to alleviating the suffering of people, I guess, which is the easy part with medicine, is that you can at least have the ethical imperatives that should guide that when we think about what the analytical framework is and what the categorical framework for people are within that. So that's not really clear, but that's what
Okay. So when I was trying to read that book, something I felt was that the tone was not super friendly, at least in the beginning part of it. I felt like it was slightly abrasive. So that was maybe one thing that started to make me think that I didn't want to read the rest of it. But then another thing is I pulled up her TED Talk, and kind of felt like it was that same way. And the comments below, I mean, probably shouldn't judge too much on comments, but the comments below were kind of going along maybe with what I was feeling about the tone of the book, or I felt like it wasn't too scientific as far as the discussion about anyway, I won't get into it too much, but it didn't seem very scientific to me. So that added to me not wanting to read the book. And then I had one other good point I wanted to make, and it's evading me right now. But anyway, the other point is she kept mentioning race-based medicine over and over. It was like her key phrase, and she was railing against race-based medicine. And I'm like, I've never heard that term before. So I did a Google search for race-based medicine, and the top 3-4 hits were all her stuff. So she's either just a major person trying to bring light to this, or it seems like kind of her catchphrase that she was trying to popularize, I guess. But you know, talking about race and race relations is kind of difficult because people can often take you wrong. Maybe I was misunderstanding what she was trying to say, and it turned me off. Anyway, we can get back to this book unless someone wants to say something else. But
I would just say that I think she's making arguments that if you're familiar with the social sciences, she's making arguments that are very strong from a social science standpoint. She has a lot of citations to studies that are looking at, and it's hard when you're looking at culture. So my husband's an anthropologist, and so he has to, and he's very strong in both quantitative and qualitative. But it's not as straightforward as it is for a laboratory scientist. So you have to try to find ways to design studies to get at complex questions that have to do, like if you do a survey, you have to think about what's your language? Are people going to interpret your question in the same way? How are people going to think about this word? And so the social sciences work towards study design and quantitative statistical analysis to test their hypotheses, but it looks different than I'm used to seeing. And I think that can be a turnoff for people. But I wish it wasn't, because I do think the social sciences have a lot to tell us that's really important for medicine and genomics and how we're moving forward with this. Yeah, if you have a chance, Joel, I'd encourage you to try to sit with it again. I think she's laid out her argument really well. And if nothing else, it might be worth getting through the book to see what the totality of her argument is, and then you can decide like the parts that you agree with or don't agree with. So yeah, thanks for bringing that up though. I don't at all feel like you derailed us, because I think that was, we had our discussion last month, but it intersects with this. And I do think to the larger theme of talking about genomics, I think that's something that's worth discussing, and that might make it into the classroom or not, depending on how people choose to teach this. So I think it's great that you brought it up. You shouldn't feel like that was a diversion at all. But I see that from the time, I don't want to keep people too much longer. I know everybody is probably busy with many things, including getting ready for the semester. So to wrap up, I thought I would see if anybody has any final thoughts about this book. Joel, I forgot that you suggested this, so I'm glad you reminded me that you suggested this. I liked it. I definitely see Sangeeta's point that the beginning of it, and Joel's point as well. I think you're both right that the beginning of it is starts out kind of baseline level, basic information. But the last couple of chapters especially, which we talked about, really get into looking at ideas that you would associate with a society of genes, how they cooperate, how they're managed, the different hierarchies of roles, and then things like cheats and freeloaders, which we didn't get to, but the lines and signs, and how their entire purpose in the human genome is to just make copies of themselves. So I feel like it sets out an interesting thesis. I think it's a worthy companion piece to The Selfish Gene, which I read a long time ago. But I'm glad you suggested it. I'm glad we read it. And I think I will be using, at the very least, I think I'll be using chapter 1 as a refresher course or refresher session for students in genomics. And at this point, I'll turn it over to see if anybody has any final thoughts about the book.
I think, Laura, you also mentioned this on Twitter. This is one thought that I had, that the use of the purpose-driven language in the book is quite strong, and they do not caution against it often enough. Like, for example, if you read The Selfish Gene, Richard Dawkins keeps cautioning about it over and over again, so that you can never forget, because this is a very dangerous territory when people start misunderstanding that. So if you are, I think you might want to remind your students that we use the language loosely, but doesn't imply purpose.
I mentioned that I read the comments on the Dorothy Roberts video, but I also read Amazon reviews and things like that. And so when I was looking at this book as something maybe I'd want to nominate, I was reading the reviews, and for the most part, it gets good reviews. And I do think it's big. Those good reviews are from people that read all the way through. But anyway, the one bad review that I read about it was complaining about how many analogies and anthropomorphizing, making too many analogies and metaphors out of the book. And but the title of it is The Society of Genes, and so it's already a framework where you have to be open to thinking about genes as compared to a society of people or things like that. And they also even had 2 pages, maybe 1-2 pages, and towards the end of chapter 1 where they did a disclaimer on all the analogies. But anyway, if you're one of those people that is persnickety about never talking in analogies or metaphors or anthropomorphizing, someone's got to say that for me, because I forget how it goes. But anyway, if you've got a problem with that, this book probably not for you. It'll probably grate against you. But as far as a new way to think about evolution or whatever, I think it's pretty interesting.
Okay, so it's interesting that you bring up the anthropomorphizing of the Society of Genes, because I also found that a bit disorienting, but it's also weird because this comes simultaneously to me currently taking a continuing education course where the textbook is entirely about student engagement techniques. So the reason that the Society of Genes was completely novel to me and made me kind of flip a lot of things in my head around, considering that I will be teaching in a few months, is this is the first time I've ever seen a science book that has centered presenting its mental models in advance of breaking down the science. It's a very inverted framework for a lot of us who teach from more traditional models of textbook and transmittal models of education, which we're trying to move away from with active learning strategies and paradigms. But this is also considered a central tenet of effective science communication, which is creating your engagement with an audience through mental models that sometimes are appealing to aesthetics or emotional investment, which is something that we in our scientific training never really talk about. We talk about how to communicate in the context of, our journal articles, and even though that's technical writing, we still even avoid calling it that because it's supposed to be efficient and schematic. And in terms of our teaching, because of our training, I think we're more embedded in our accuracy and in this holistic thing that we've come to understand in great detail in terms of the compartmentalizing that each of the chapters and their very picturesque and narrative-driven outlines that you can throw your mechanisms of genetic strategy and regulatory strategy and diversification step on. Because this book is I actually found it exhausting, because it was in a good way, because it's also storytelling. These are, the Moshev thing is really a narrative, and it's just like, it's basically a parable to tell you how diversification happens in the Society of Genes. And it's very, very different, I think, for a lot of us who's come up through highlighted box paradigms and bolded text paradigms and the little box that shows you, ball and stick models that never structurally deviate too much from the biological process that's going on. So it's a good challenge to think about how you educate using the vehicles that the authors have made decisions about using. And I actually wish I'd read the before and the after before I'd read the middle of the book. So the prelogue and the afterword, because they actually went ahead and described the connectivity between the chapters, which is a really good orienting tool to have, especially if you're thinking about teaching with the book. So that's all I have to say.
Yeah, I think you've all made excellent points. I'm glad, Sangeeta, I'm glad you brought up the idea of I had a professor in my freshman biology class who would just shout anthropomorphosis and teleology, and you'd be like, oh God, you know, so you were never supposed to explain anything with either of those things because they were the worst sin you could commit. And so again, I actually wrote it in my notebook in like capital letters, quoting my freshman biology professor. But I think to the point that everybody else has discussed, you kind of have to decide to what extent are you going to go down that road to help illustrate some concepts for your learners or your readers in this case. And I think you have to walk a very fine line where you don't leave your audience thinking that there's purpose behind natural selection, evolution, cancer, you know, that the cells and the genes want something. So if it's shorthand, I think what Joel mentioned, I think, is that you've got to be very careful to make sure that you're always circling back to say, this is shorthand, this is shorthand, this is shorthand. I think they did have their little bit where they said that, but they kind of said it once and then was like, but we're all sure you guys know that it's shorthand. But if you're not familiar with this and you keep seeing it presented in that way, it can like worm its way into your mind. So like, oh, well, the genes want to make it to the next generation and they want to survive and they want to outlast their competitor cells and all this kind of stuff. So I do think that that's tricky. I do think that's tricky. But Nicole, I think you're right. I think it really adopts this interesting narrative style. It's very conversational in the way it's like, okay, so say it's like this thing. I mean, you get a story about Bob Marley's toe, you know, it's like, oh, okay, that's random. So it's an interesting, I'm happy to have read it right before teaching genomics because I think it offers some, I'm definitely gonna keep it on my shelf and probably go back to it a couple times because it offers some different examples and some different ways of illustrating what are some really important and fundamental theoretical concepts that I think it's good for students to attack from different angles. So I could certainly see myself combining this with some other resources as another way of getting students to think about a lot of these topics, which is really, I guess, what everybody is always trying to do is attack things from multiple ways to try to get students to familiar with and to understand a topic. But any other final thoughts? Okay, so thanks very much. I think this was a great discussion, especially being so close to the end of the summer, beginning of the fall. I really appreciate it that, that folks are able to join us. So what happens now for MicroJC is that in the academic year, we turn back to our regularly scheduled programming of being a journal club. So I will be, hopefully it works out that we'll be able to do a journal club in September. So this is a regular journal club. I take suggestions for papers. It's generally within microbiology. We kind of have in the past tended towards certain topics because they're recurring and returning people's interest, but anything within microbiology I am happy to tackle. Kendra and Nicole had suggested a couple of things that were outside my wheelhouse that I'm glad I read. So I'm always happy to read stuff that are that come from a wide range of things. So if there's something on your to-read list and you want an excuse to read it and talk to people about it, absolutely send me an email or a tweet and I will be glad to put it on the list. So I don't have any ideas for September just of yet. So if there's a paper that you're really eager to read, then feel free to suggest it and we will try to get that into our queue. So with that, I'll thank everybody for joining us, and it's been a really good summer. I've learned a tremendous amount about genomics, which is good because I have to teach it. I have taught it before, but you know, I'm refreshed and ready to go. And I really appreciate everybody coming along and reading these books and contributing to the discussion. So thank you very much.
All right. Thanks for hosting this.
Yep.